So good morning, or rather good noonish. Uh, any questions from yesterday or day before? Okay. So since I don't see any raised hands, so anybody is speaking, so I assume that everything is clear or nothing is clear. I hope it's the former. Okay. So I continue the discussion that we have had. So I started with discussing with you the evolutionary forces, and I said to you about genetic drift, and have been focusing because that's the title of these lectures is Introduction to Stochastic Modeling. So therefore, we have been dealing with. I've been discussing all this so much about the genetic drift. And as I said, that the running thread through my lectures would be this Leventin's paradox, which we discussed yesterday, which was the question was simply, let me remind you, was that why is neutral genetic variation, why is, which I'm denoting by H, why is it roughly independent of census size? Okay. And then the idea that has been around for some time is that somehow we should not think of census population size, but rather an effective population size, NE. The idea being that there are other processes that are going on, as a result of which the population which will contribute to the genetic variation, the succeeding generations, is smaller. Typically, it's smaller than the census population size. And that would allow us the data to somehow move in the Kimura's prediction, which you remember is 4 n mu. Okay, so more properly, perhaps 4 n e mu. And there are several possibilities that people have, mechanisms that people have proposed, including demography, mutation rates, dependence on any, and then selection at linked sites. Okay, so we discussed briefly about demography. I said very little about mutation rate, but then this is the one which is sort of very important and also very challenging. So we started to discuss a little bit about selection yesterday. And I want to say a little bit more because it would be relevant to what I want to say next. We haven't said anything about linkage yet. So I want to discuss that a little bit about, I'll say the basic stuff about recombination also. And then we will put things together. And I will very briefly say about what are called sweeps, selective sweeps, and of various types. Okay. So I was planning to do more detailed stuff, but I think maybe it's best for this audience if I just keep it to a simple level, and then at some point I will just stop. Okay, so so we discussed about selection yesterday, and we discussed about fixation probability, and we discussed in some detail that it has this formula due to Kimura. So for a single mutant, initial mutant, it is simply given by this. Okay. Today, I want to discuss a little bit more and still about selection, and I want to discuss about mean fixation time because it would be relevant to what I want to say next. And these fixation times are very difficult to calculate analytically, at least, and they are generally challenging problem. But they are very, very important. I mean, we'll see that how they enter the picture. I'll give you one example today, and I'll give you one example next week as well. So let's at least try to understand them a little bit. Okay, so we want to discuss a little bit about fixation time. We did this discussion for neutral one already, and I want to now discuss about when we have selection. One way, of course, is to use the diffusion theory. Okay, so we can always use diffusion theory. Not always. I mean, in many cases, we can use diffusion theory to understand this, but it's just too complicated and it's not particularly intuitive. Okay, so you can sort of, run through the math, but it doesn't give you a nice intuition. However, it's possible to do something simpler if I have strongly beneficial mutations. Okay, so it's a somewhat simpler theory. It's not a diffusion theory. It's something which is between deterministic and stochastic theory. And therefore, I will call that as a semi-deterministic theory. So ideas are simple and it's nice to know them. And that's why I'm going to sort of describe them to you. So first of all, remember what is strongly beneficial. So some of you were asking this question yesterday. What is weakly beneficial or strongly beneficial? Strongly beneficial simply means that this product NS, if it is much, much larger than 1, NS is 100, 10 or something, I'll say that it's a strongly beneficial mutation. If NS is smaller than 1, okay, then it's weakly beneficial mutation. Okay, and that's what this weakly or nearly neutral theory was about. That's where the nearly neutral theory operates. All right. Okay, so we remember this from yesterday. Okay, so now for the strongly beneficial mutations, some simplifications occur, and I will show them to you on this slide. Okay, so what is shown here? What is shown here is the mutant frequency as a function of time. And I'm showing you 3 different trajectories, where in each trajectory, we start with a single beneficial mutant. So that's what the point over here is. Okay, the frequency is 10 to the power minus 4. And as time goes on, we are looking at only those trajectories which are fixing. Okay, so I'm not showing you anything which goes away, loss occurs. Okay, so we are just focusing on those ones because they are relevant to the discussion. So we start with a single beneficial mutant, and because there's stochasticity, these 3 trajectories, they look different from each other. But about 100 time steps or so, you see that all 3, they seem to behave in a similar way. I'm not saying that they're identical, but they are more or less same. Okay, that means the fluctuations that we had in the beginning at largish times, when the allele frequency has become substantial, here more than 0.1 or so, that means you have 10% or more of the mutants in the population, then this suggests that for strongly beneficial mutations, I can neglect stochastic fluctuations. Okay, so initially we have a stochastic phase. Then for the mutant frequency, when it has become large enough, then I can neglect stochastic fluctuations. And in this case, this becomes fixed. This is not a complete story, because you see, this is the mutant which is fixing. But if I look at the complementary quantity, the wild type frequency, so wild type frequency, so here is again the mutant frequency, which is sort of, there's some jiggling, something, and then it looks sort of smooth curve, single trajectory is what I'm showing you now. So it's jiggly stuff, and jiggles are less, it looks very smooth, and then it becomes 1. Correspondingly, the wild type frequency, it's smooth, in the same time duration, it has less jiggles, that means less fluctuations. But at larger times, when the mutant has fixed, at that time, the wild type frequency has become low. Its frequency is like 0.01 or so. And that time, the wild type frequency is evolving stochastically. Okay, so for strongly beneficial mutations, which we have sort of a nice picture, it says that at short times and at very long times, when the fixation is just starting and fixation is about to end, the allele frequency dynamics are stochastic, and in between, fluctuations can be neglected. So we can divide these dynamics into these 3 parts, 2 stochastic phases, and these are in between is the deterministic phase. Okay, so deterministic theories are relatively much easier, actually. Most of the time, they are much easier to handle than the stochastic one. Stochastic things are kind of pain, but we need it, of course. But, if I could just handle only deterministically, think that would be nice, right? So let's give it a shot and see what happens. So first, let me just say, for completely neglect stochasticity. And I want to see what I can say about fixation time. Let's neglect stochasticity completely. Okay, so fully deterministic theory. Deterministic means no genetic drift. Okay, only selection. So when we have only selection, we learned about this yesterday. So remember what I said? I told you about something called logistic equation. Again, it's a very, very standard thing. So let me just remind you once again. So if X is the frequency, the mean frequency of mutant at time T, then I told you yesterday that it obeys this logistic equation, very famous standard equation, not just in genetics or something, it's just, in all sorts of places you will find it. The S was the selection coefficient of the beneficial mutation. Okay, so this from elementary school calculus, you can easily solve it. Okay, and then using which you can show that X of T, it looks something, has this solution, which I will just write it down for you. 1 plus 1 minus, sorry. So it's 1 by 1 plus the initial frequencies, the ratio of the initial frequencies into e to the power minus st. Okay, so a simple formula, you can show it. All of you actually can show this. It's not difficult. So suppose I just neglected stochasticity completely and I said that I start with some initial frequency, and this is how the allele frequency deterministically will behave. And I'm just asking the question, how much time would it take for X to become fixed? That means that X to become 1. Okay. Looking at this formula, I'm sure some of you are very comfortable with these things. Can anybody say how much time would it take for this object to become 1? Any guesses? Just give it a try. No? Okay, so I'll give you the answer. The answer is it will take infinite time. You have to put t to be infinity here, then only X becomes 1. Okay, so it's saying that if you have, it will take forever for this population to become fixed. Does that make sense? Anybody want to say something? Yeah.
Yes. It makes sense.
It makes sense. It will take forever.
Yes.
But the thing takes forever. I started with only 1,000, a population of size 1,000, in which I have a single mutant, and I'm asking how much time would it take this mutant to go through the population? How can it take forever? If I have finite things, so this is a sort of a simple abstract, not really abstract concept, but it's a simple thing, right? If I have something which is finite, okay, finite things take finite time to happen. I have a finite population, and this single guy has to, single mutant, I started with n minus 1 A1s and 1 A2, and all of them have to become A2. There are only n of them. Okay, it cannot take forever. The time is also going to be finite. The reason this theory is giving me this kind of absurd answer is because deterministic theories assume that your population is also infinitely large. So in itself, it's consistent. That is, if I had indeed an infinitely large population, there are infinite number of A1s in which I put a single A2 and ask how much time would it take for all of them to become A2. There are infinite number of A1s, so they will take infinite time. Okay, but we are dealing with finite populations. All realistic populations, all real populations are finite, right? So therefore, if I am using this deterministic theory, it will give me a wrong answer. Okay, so it's a sort of basic point that, just if I just neglected stochasticity, I mean, so I was hoping that I could do something simpler, which is that, get rid of drift and I'm home. But unfortunately, that's not true. Okay, so deterministic theories, pure deterministic theory will not work. Okay, fine. So then we have to go back and try to do a slightly better job. Okay, but without going to this full-fledged diffusion theory. So that is what I'm trying to convey to you. So let's rewrite this one. here d by, so this is our logistic equation. Okay, and this I'll just rewrite this answer to the solution of this, that if I want the frequency at time t to be x, then it can be rewritten as, I'm sorry, I'll try correctly. So let us rewrite this as, if I say that TF is the time at which frequency becomes XF. It's just from here. I mean, I'm not doing anything great here. It's just school level calculus. Okay, nothing very fancy. So it's simply given by this formula. And I'm just rewriting it simply because it helps me to see things clearly, more clearly. Okay, so it's just the solution here is simply, can also be rewritten like this. So let me just say that, okay, fine, my attempt to just deal with things deterministically has failed. So I'll try an attempt to, in which I try to make the theory more sophisticated and say, okay, fine. Okay, I know that there is a stochastic phase, at least initially. And let me just say that I'm not going all the way to fixation. For simplicity, for demonstration purposes, let me just say that ask for a time in which the final frequency becomes 1/2. Okay, so our LE frequency is doing this jiggly stuff, and it reaches somewhere 1 here, right? But let me just ask for the time to reach 1/2. Okay, so frequency and time. Okay, so XF is 1/2, and therefore 1 minus XF is also 1/2. So XF is 1/2, and your X0 is, however, 1 by n. We're starting with a single mutant in a population of size n, right? You can plug it in, it's very, very easy. And you can see that T of 1/2 is therefore simply 1 by x log of n. Okay, so therefore.
Now, what is the operator in the ln function, xf by x0 in plus my, what is it?
What is the log?
Xf by x, no, xf by x0, is it after that, what do we?
Yeah, it's 1 minus x0 divided by 1 minus x. This is the
okay, it's just a product.
Mutant frequency and ratio of the allele, the wild type frequency. Okay, good. Okay, so this gives me at least something finite. So that means I cannot get rid of stochasticity. I have to take care of it. At least it's finite, right? It was not infinite like what we got earlier. This is not quite correct, however. Yes, Shubha.
So in this particular example, you have chosen the frequency at this particular time point. You've chosen the frequency of the mutant to be 1/2, right?
This is the final frequency.
The final.
So it's fixing, but I can. Nobody said that I cannot ask the question that when it reaches frequency 1/2. I can ask that question, right? It can reach 1/2, not necessarily 1. I will come to 1 also in a minute. Okay.
Okay.
Okay. So all I want to say here is to demonstrate it that if I took this little, I did not ignore the fact that there's a stochasticity and which I am putting by an initial condition here, that I'm starting with a single mutant, then I just get this answer, which is log n by s. Log is a slowly decaying function, but whatever, a slowly increasing function, but whatever it is, the answer is finite, unlike the previous attempt. However, this answer is not really correct. One thing is that, I've been emphasizing to you that in these theories, there are these two scales, the drift and selection. N does not come alone. It comes in combination with mu as n mu, as we saw for heterozygosity or the pairwise diversity for n mu. It does not come alone and also comes with S, right? Like we saw yesterday for the fixation probability. So this formula itself is, not quite right. And the reason is that I'm assuming here, I'm looking at those trajectories, okay? Those trajectories that actually go to fixation. Remember, even if we have a large population, but finite population, there will always be trajectories which are going to go extinct. when I'm saying to you, look at a subset which is going to fixation, we have to factor in this fixation probability. Okay, that's why this answer that I just gave you is not quite right. And this not being right, it's not just a minor quibble that, yeah, let's put not N, let's put S with it or something. It's actually a very important factor. It plays an important role when we do things properly. Otherwise, things don't quite work out. Okay, so I have not demonstrated that to you, but it's really important to take this fixation probability still into account. Okay, so then if I start with so I'll give you a simple argument. If I start with a single mutant, it fixes with a probability 2s. 2s, remember, if you like Kimura's formula, under strong selection, we get Haldane's formula, right? Very easy to remember. The fixation probability of a strongly selected beneficial mutant is simply twice its selective advantage. Okay, so if I start with a single mutant, the chance, so this is the number of mutant, initial mutant, and this is the corresponding p-fix. So if I start with a single mutant, it fixes with the probability 1/2 with probability 2s. So therefore, if I start with 1/2s number of initial mutants, they will fix with the chance 1. So the fact that I'm looking at these trajectories only that are going to fixation, okay, so the ones, if I'm focusing only them, they're not getting lost. They are definitely fixing, right? So when I'm neglecting this part, one way of to take care of that is to say that we are actually not starting with one mutant. It's as if I'm starting with larger number of mutants given by 1/2s. Okay, so that's the sort of idea that you have to think a little bit. It's also to do with something called conditional probabilities. If you like, Sachin will explain that a bit more tomorrow, but this is what basically one is thinking. Okay, that because of these losses, it's as though we are not starting with a single mutant, but a larger number of mutants. Remember, S is smaller than 1. Okay, so therefore, my x0 is not 1/n. Instead of that, if I start with x0, that means because the frequency with 2ns, okay, if I do that, then I get this p½ to be 1/s log of 2ns. And all is good in our mind, in the sense that I told you, n doesn't come by itself. So that is what you have to factor in here. So you have to make this theory a little bit more sophisticated by just using these simple formulas that we had discussed earlier. We can make another attempt. Yeah, go ahead, Shubha.
Kavita, I didn't understand how if you had 1/2s number of initial mutants, it has a probability fixation probability of 1.
Yeah, this is just unitary method from school. One
okay.
fixes with 2s. Yeah.
Okay.
Sorry, I should have explained better. Yeah. Okay. So therefore we have this. And now I can go ahead and do an attempt 3 and say that, you are cheating a bit. You are saying that we'll go only halfway. I want to go full way. That means I want to do full fixation. Okay. But if you look at this formula, that means if you put xf to be 1, there's a problem because this will become 0 here, and due to log infinity, it gives rise to problems. So I am quite okay if I don't become, the frequency is not exactly 1, but teeny bit smaller than that. That means if I have million individuals in the population, and I'm happy if you tell me the time to reach million minus 10. Okay. So even million minus 1, doesn't matter. Okay. So if I put xf equal to 1 minus 1/n and x0 to be 1/2ns in our simple deterministic theory, we are getting into a problem. It gives me, let me write it here. It gives me this t fix to be 1/s log n square s, which is not good. The combination which is permissible is n times s. If I have an n square s, then I better have n square s square. Okay, so this is also not really correct answer. How to fix this, to take this full, yes, Shubha. Go ahead,
Yeah, I
Shubha.
have to lower my hand.
Okay, okay. So, yeah, Pritish.
I just had a question. Like, how do we know that we are on the right track only if n and s come together with the same power? Like, why not n square s anywhere?
Yeah, so that's the character of this this diffusion theory. Since you asked, I'll explain that to you. In fact, okay, so I'll just tell you. So this has to do with the limits that we take when we write down those theories. The limits is that we are thinking of large n. I don't know if you have sort of a more mathematical training, but anyway, I'll tell you. So the assumption of these diffusion theories is n is large, infinite, not literally infinite, but pretty large. And the selection coefficients are small. Something is very large and something is very small, such that this product remains finite. So it's a limiting procedure. If you don't do this limiting procedure, then the theory, those equations break down. And in fact, I probably have it here. So you see here we have 1 by n, and here we have a s. S is small, n is large, so therefore 1 by n is small. And these two terms have to be of the same order. If I put n square, then 1 by n squared, then that term will be smaller than this. So it's some logic of that sort is what we have to use here when we are saying that this product has to be n times s, both have to be linear and not n square s or ns square or something like that. Okay, so this you probably have to think a bit more, but that's basically what goes in here. I'm not sure if that answers your question, but
yes, ma'am, it does.
Yeah,
It does. Thank you. Thank you. Yeah, thank you.
Somebody else had a question, I think.
Can you please explain why are you starting with xf is equals to 1 minus 1 by n for this attempt 3?
Yeah, that's because I can start xf equal to 1. Still haven't gotten hang of this. If I start with xf equal to 1, which is what you want for fixation, right? But what will happen if xf is 1? Then this will become log of 1 by 0. 1 by 0.
Yeah. That I.
Yeah. Okay. That's okay.
Then
So I'm saying why 1 by n? You can start with 10 by n. No issue.
Okay.
It's just that this number should be much more smaller than n. That's the thing. So C should be much more smaller than n. That's all.
Yeah.
Okay, and you still get this problematic answer. Okay.
Yeah.
Good.
Thank you.
Yeah. Okay, so one can do that, but so therefore this is a simple question, and this is some fixation time, probably the time of fixation. Of course, you can go to the full diffusion theory, and there are this things called double integrals, and they are horrible. Actually, I mean, horrible means you can do it, but they are not very nice objects to handle. You can handle it if you just sit, and do it. And therefore, it's nice if you can sort of build up some sort of more intuitive understanding of this. So in that context, there has been some more progress where we can take both the phases into account. So I will not do more details of that, and I will simply refer you to some recent work. So there was some nice work by Guam Martin, who will speak next week, and Amaury Lambert. So they have done some nice work which allows you to actually calculate not just the mean fixation time, but for the distribution of fixation time. Remember, fixation time is not just a number, right? It's not just mean. There's a whole distribution of it. So they were able to find some approximations for it. Okay. And so with Sachin and I, and also we have also been able to extend it further. So, but it's a bit more technical. So unless somebody is really interested, I'm happy to speak to you separately, but I'll skip it otherwise. Okay. So the only good news, if I want to convey to you, is that you can, at least for strongly beneficial mutations, one can sort of build up a simpler theory, simpler than diffusion theory. One can understand, okay, can be built, which is somewhat more intuitive. At least I find it more intuitive than diffusion theory. Okay, okay, that's one point. And the
Kavita,
yeah, go ahead, Bhavya.
I was wondering whether, I was wondering whether your selection coefficients can be a function of time.
Yeah, well, that's what you're going to hear next week. Yes.
Okay.
Yeah, in general, selection coefficients can be a function of time. Population size, we have already heard, are a function of time. So those are the more general situations, right? And therefore, that's the work of Sachin in his PhD. And I'll speak about that a little later. So all this edifice that I'm building in these 3 lectures is finally to come to that situation, which is even more complex. But I hope you will be able to appreciate that once you know the simpler stuff in which everything is constant in time. Okay, good question. Okay. Okay. So, however, I want to still keep ask you to remember this approximation, because it's sort of, although it's for half time and so on, but basically it's sort of correct. Even if the t half, even if it is full time, is more or less correct from the more, you know, detailed calculations that we can do. Still, this roughly turns out to be correct. And that's again another point. So if you're somebody who likes to do analytical work, it's nice to have these kind of approximations and have these intuitive theories, etc. But it's also important to have something which is, you know, more solid, so that you can compare things and ask, are these approximations really good? Do they actually work? Okay. So yeah. Okay, fine. So I'll just box it to, because I'll be coming back to it. Okay. So we will keep this in mind. I saw one more point I want to make here. So I spoke about strongly beneficial mutation. What about deleterious mutations? Can I understand something about their fixation time? So of course, for deleterious mutation, you expect that if I start with a single mutant, most of them will get lost, right? I mean, very, very fast, very quickly they will. But once in a while, some of them will sweep or fix, right? And for deleterious mutation also, I can ask this question: what's the mean fixation time? From this more detailed diffusion theory, there is this amazing result. I'll tell you. Oh. Okay, how do I get rid of this guy? Okay, so there's this result. I'll just take you in words. It's called the Maruyama Kimura symmetry. Kimura you've heard a lot, right? So Maruyama, I'm not sure if it's a student or something, somebody like that, but there's a symmetry which says the following. It says that if you're looking at only those trajectories in which fixation occurs, then the mean time for fixation of a deleterious mutant with effect size S, or rather minus S, is same as the corresponding time for beneficial mutant with the same effect size. So here mean is provided. Of course, we are looking at those means in which we are looking only the fixation ones. So for again, the experts, properly, the right thing to say is the conditional mean fixation time. Okay. So this is a sort of very, very difficult to say this from intuition. So when you have this full-fledged theories, they may be, challenging analytically to do things, even to read those papers. It can be very challenging. When you start, it's very difficult to understand them. But to come up with this kind of symmetry, to understand the meaning of the symmetry, to guess that the mean time would be the same, whether you're a deleterious mutant or a beneficial mutant. That's very hard. So that's one reason I like to have these kind of more sophisticated theories, because not everything can be guessed intuitively. So just to be sure that you understand what I'm saying, I start with single mutant, forget these guys, and this time in which it actually fixes, it's the same whether this mutant was deleterious or beneficial. Okay, and this is not simply just some mathematical statement, so let's not bother. Actually, it has a consequence. So I'll come to that in a minute. Okay, so just to keep this in mind. So let me just box it again so that we remember. Okay, fine. Okay, so
is it because the deleterious mutation is linked with the beneficial?
No, right, no linkage. We are thinking right now, just single locus is what we are discussing. We haven't gone to linkage as yet.
Okay.
Okay, so we will come to that, and the consequence I'll describe at that time. Okay. Yeah. So now, since you are, yeah.
So it's slightly hard to understand. It takes the same amount of time. The probability is much lower for deleterious, but it still takes the same amount of time. I don't know how.
Yeah. So therefore, one can argue, so the one argument, and these arguments are sometimes quite of shaky, meaning you can always come up with another argument. So I would have thought that, because the deleterious mutant, it should take longer, right? Because, this guy will have more fluctuations. Neutral mutations and deleterious mutations, you cannot do anything deterministically. Okay. For the beneficial ones, you can, but drift would be very strong is extremely important for deleterious mutations and neutral mutations. There's a lot of work with advantageous mutations on adaptation, where you can, to some extent, you can neglect drift, but for deleterious and beneficial, deleterious neutral, you cannot. So I would have thought, because it's a deleterious one, it will be subject to so much, drift, and will take some time, this one, that one, and whenever it fixes, maybe it will take longer for it to fix. On the other hand, you can argue that, look, it's deleterious. If it hangs out in the population for too long, then drift will bring it down. Okay. So therefore, to escape extinction, it better fix sooner. For beneficial mutation, you expect that it will fix pretty soon, right? Because it's good. It will rise quickly in the population and fix. So one argument for the deleterious mutations is similar in the sense that to avoid extinction, okay, so I'm not a good mutant, so I better spread soon. Okay, so from that logic, so the argument using which one tries to rationalize it, that for the beneficial mutant, it's the growth is fast, and therefore you can expect a shorter fixation time. Deleterious mutation also, for the same S, will fix soon, but that's because to avoid extinction. Because if it is there in the population for too long, and it's sort of still in the intermediate frequencies, drift will be strong and it will bring it back down. So if it's going to fix, it better do it soon. So that's one way to rationalize it. Make sense? Or I'm sure you can come with some other argument, but it's a little difficult, to understand intuitively. Convinced? Okay, but do think about it.
This is probably very naive, but if we look at the sort of correct equation that you talked about in the previous page, and again, if we just say that a deleterious mutation has a negative S, again, this is very probably naive, but then the negative signs sort of go away, and mathematically it makes sense.
No, well, log of minus ns doesn't make sense. Minus s doesn't make sense. The negative sign inside.
Oh, right. But so then if we just take a mod,
we'll give you imaginary time.
Right.
Yeah. So what you're saying is sort of right. So you have to replace it by the modulus of that.
Yes, exactly.
Yeah, so that is true. But I mean, I cannot build this argument for the deleterious ones because you see, there's a problem. Hit this guy. Should I just wait or what? Okay, so the problem is this. If S is negative, dx by dt is negative.
Right.
Which means deterministically, deleterious mutation will go to 0. There's no way it can spread.
Right. Right.
That's why I was emphasizing to you for deleterious, and similarly for neutral, dx by dt is 0. That means there will not be any change in frequency. So since you asked, let me just plot it for you. If S is positive, that means for the beneficial one, this equation will predict that you can fix. If it is deleterious, it says you will definitely, okay, even if you start with a high frequency, it will go to 0. But if it's neutral, it will not change. So fixations cannot be captured here. So the point I'm saying is that you cannot, use this argument to come to this answer, but answer eventually comes out right because of the symmetry.
Sure. Okay.
Okay, thank you. Anything else? So this is kind of very amazing. And actually, in fact, I'm embarrassed to say that we didn't know it ourselves for quite some time. And we discovered in the literature recently in the context of Sachin's work. But I find it quite amazing. Yeah. Okay. Yeah. Okay. So let's now go to recombination. So because I want to discuss about sweeps, so therefore the other things that so I'm done with the basic discussion about the selection. We have discussed fixation probabilities and fixation time, and I now want to think about linkage. So let's now talk about recombination. So yesterday you heard a bit, but I now want to see what recombination does in some simple model. Okay, and it's best demonstrated if so for recombination, I cannot, of course, have single locus. I have to think of at least 2 loci. Okay, so far I did not say it explicitly, but as Bhavya asked, I am all this time, or mostly, I am thinking of only single locus. But now let's come to 2 loci, because then only recombination can occur. So in the first locus, let's say we have 2 alleles possible, A1 or A2. And in the second locus, we can have B1 or B2. Okay. And for discussion, we'll again, the usual approach, neglect everything else so that we can understand what recombination does alone. So no drift, no mutation, no selection. Okay. Just to understand what drift does, what recombination does. Okay. And I want to know, for example, so how many genotypes can we form? Well, there are 4 of them, A1B1, A1B2, A2B1, and A2B2. Okay. So there would be matings between these. So for example, A1B1 can mate with A2B1. So the way I understand recombination is that, these loci, they will break apart, the 2 loci, and then there will be a genetic material swapping. So let me explain what I mean. So just a break. Okay, so now they are here, and then you swap the material. So then it will give you, so this is not a good example. So let me say it again. So suppose I have A1, B2, and I cross it with A2, B1. Then, first of all, there's a break point here. There's only 2 loci. So A1, B2, A2, B1. And then, because of recombination, it's possible they swap the material, giving rise to offspring like this or this. Right? Okay. I'm sorry. A1, B1, and B2, B2. Okay. So these are the sort of matings we want to think about. That how would it change the frequency of these 4 genotypes? I'll write one of them, and I'm sure you can write the other ones. So let me write down, how does this frequency of A1, B1 change because of recombination? How can it change? So A1, B1 can be produced That means its number can increase. From what kind of mating? So I'll write one for you and tell me if you agree. Suppose we have this mating, the one that I wrote, in fact, right? This mating will produce A1, B1 for me, right? And therefore, there will be a positive change. That means there will be increase in the frequency of A1, B1 because of these matings. So these two mate, and that means recombine with the probability R. But both of them have to be there. So you see, you have a bilinear equation. You have to write both of them. Okay. Anybody wants to tell me if there can be matings which can result in the loss of A1, B1? So you understand that how A1, B1 will be produced. How will it be lost?
If A1, B1 mates with A2, B2.
Correct. The rest will not do anything. They will not change. Right? So when these two mate, they'll produce other things. A1, B1 will be lost. Okay. So you can rewrite all this stuff. Similarly, if I were to ask about, I guess, which one will it be? A2, B2? Yeah. So this would be same. Right? It will be basically all is the same terms, right? But if I write for A1, B2, then it's just this R times frequency of A1, B1, A2, B2 minus, give me a second, Pratyush. A1, B2, A2, B1. And similarly, you can write for the A2, B1. Okay, that will also be the same. Yes.
My question was, so are we assuming that recombination always happens in this case?
So there's a chance. There's a chance. So therefore, both are there, and the probability that it happened is R, or rather the rate at which it happened is R.
Right. But like if in situations where the parental combination is retained, that will also, that does not change the frequency, is what we're assuming.
Yes.
Okay.
Yes.
Yeah,
Not assuming, we are writing equations for the change.
Right. Yes, I understood. Thank you.
Okay. Okay, so I think I've ran out of the slides. Okay. So, but let's retain this combination. Okay, let me open the new one. Okay, sorry, I lost that one, but I hope you remember. So let me just rewrite it anyway for you. I'll write one of them only. So R times this minus the loss. Okay. So this quantity is what was coming in the previous ones that I showed you. The same quantity is coming with a plus sign or a minus sign. Okay. And this is a special quantity. This is called the linkage disequilibrium. Okay, or LD, generally denoted by D. So this D, it's the frequency of these two heterozygotes minus minus these other two combinations, right? This is not very intuitive to understand, like what is this LD? By simple algebra, however, you can show that this object can be rewritten as, for example, there are many ways of writing it. It can also be reduced to the simple form. And this I can sort of understand the meaning of. It says that the frequency of the genotype together of A1, B2, how does that frequency differ from A1, whether it's coming in combination with B1 or B2, times the frequency of B2, whether it's coming in combination with A1 or A2. If these are independent, if there's no random associations, okay, so there's enough recombination so that A1 comes often with B1 as often as with B2. Okay, So there will be, sometimes it will be associated with B2, sometimes with B1. Okay, so then there's no, there's just random associations. There's no, these associations will go away, okay, on an average. So then in that case, if there's a large recombination, so in that case, this frequency can be written as a product of these two frequencies. If you like, sometimes it helps to think of another example, which is, for example, if you think of, if I throw a dice and a coin together, if they influence each other, okay, then I'll get an analog of this object. But if they don't influence each other, if the chance that I get a 6 on the dice and a head, then if those two are independent events, I will write probabilities to be product. Okay, so if these frequencies also behave independently, which they will, if recombination is frequent, then the linkage equilibrium is 0. Large for high recombination. So they behave like independent loci. Okay.
Now in the first expression, I didn't understand why there is the absence of A1B1 into A2B1.
What is the?
Like there could be another term, right? A1B1 into A2B1.
A1B1 into?
A2B1.
Yeah, but what will this mating do? It will create and then swap, but this will give me A1B1, right? And A2B1. There's no change.
Yeah. Okay.
It's a possible mating, sure, but it does not contribute to the change in the frequency.
Okay.
Right? Okay. So those are the, thankfully, just those combinations only. Okay. But if you have 3 loci and all, it's just horrible. So we wouldn't even try. Okay. So linkage disequilibrium, therefore, is a measure of how, whether these loci they are behaving independently or not. If there's a high recombination, they will come often enough with each other that these statistical associations can be neglected. If, however, recombination is small, for example, for an asexual population, A1 will remain with B2, right? Because it cannot be broken like that. It can be, you know, there's other combinations can come because of drift or because of mutations or because of selection, which right now I'm neglecting, right? But just because of recombination, if it's absent, then these associations will remain and LD will be non-zero. Small recombination. Okay. And when we're talking about linked sites, we are interested in this small recombination limit. Not very high recombination because they're all independent, and I can treat them as they are just single locus problems. Okay, and that helps a lot, at least for some sexual species. But many times the recombination is finite, and then we have to worry about it. Okay, so that's what I want you to sort of just know. One last thing about linkage disequilibrium. Using this equation is a simple exercise. You can show that the linkage disequilibrium decays exponentially fast. So exponential, now you have seen quite a few times, exponential decays are very fast decays, right? So over a time scale 1 by r, so the time for recombination to occur is roughly 1 by r. Okay, so over time scale of 1 by r, these associations will go away. So therefore, if r is very small, the time over recombination will occur is very large, and therefore associations will remain for longer. If r is very large, time for recombination is small, and therefore associations will go away quicker. Okay, so that's the sort of the intuition here for the meaning of these recombination rates. Okay, fine. So we will remember this also because again, this is going to be useful when we talk about selector sweeps. Okay. Okay, so I've been promising that I will speak about sweeps and so on. So let's get to that now. Okay. So sweeps is what? What is sweeps? Sweeps is like, you know, the way we think sweeps, it cleans things up. It's, you know, like a broom, right? So let me just write it here. Sweeps. So sweeping by whom and sweeping what? So we are thinking here of sweeping of neutral genetic diversity, sweeping R of neutral genetic diversity by linked selected. Okay, so this is a broom and this is what is being swept away. I've written here selected, and but it can also be linked, neutral ones. Okay, but this is a general term that people use to say what is sweeps. And the sweeps are of various types, and I will just focus on two of them. One is the hard sweeps. Okay, I'll explain what they are. Then there could be soft sweeps. Okay, I'll not even begin to discuss this. And then something called background selection. So I want to say briefly about hard sweeps and background selection because they're such classical processes and sometimes very difficult to navigate the literature. So, but I hope some basic stuff you can read so that learn here so that you can go ahead and read more things yourself. So this was just discussed first by Kojima and Schaefer. 67. And then Maynard, Smith, and Haig. In fact, Maynard, Smith, and Haig were motivated by Lewontin's paradox. So they call it a last-ditch attempt to save the neutral theory in order to explain Lewontin's observations. And background selection. So this is Brian Charlesworth. Morgan and Deborah Charlesworth. (1993). Okay, so these two are what I want to describe to you. Okay, so how is this hard sweep? How does that help us to explain how neutral genetic diversity reduces? So let's discuss that using first an example. So I will assume no mutations. And for fun's sake, let me right now even assume there's no recombination. And we have strong positive selection. Okay. There could be deleterious sweeps, there can even be neutral sweeps, but let's right now just think of beneficial sweeps. Okay. Yeah. Okay. So let me just draw a little example. So like yesterday, we considered 5 individuals. Similarly, I'm thinking of 5 individuals and they have some genetic sequence. And there are these crosses means, just like yesterday's example, remember I said if the loci are same, I will not write anything. Wherever there's a different nucleotide at a locus, I will put a 0 or 1. So that's what I'm trying to denote by the crosses. So just to emphasize that there is genetic diversity, I'm drawing different colors. So these are all neutral at these loci. At this locus, for example, these sequences are different. Okay. And but these green cross, blue cross, red cross, they're all neutral, however. Okay. Okay. So this is suppose I have these sequences. And at some point, perhaps because of environmental change, say one beneficial mutant arises. Maybe this neutral one becomes beneficial, or just a new beneficial mutation arises. Okay. And now I'm going to switch off no more mutations. Just one has come and let's look at it, what it does. So because I'm saying there's no recombination and it's just a strongly positive selection, that means it's unlikely, it is a chance of course, but if it's unlikely that this mutation is going to get lost, then because it's an asexual population, these guys will always be joined, will always be linked with it, right? Under the assumptions that I stated. So after some time, therefore, what you expect is, so you expect, this is what you will get. whatever that color is. Okay, so these are my 5 genetic sequences. So what has happened? Initially, I had a lot of genetic variation, right? There were this green ones, blue ones, red ones, and so on. But because of this linkage and because it's a selected one, it dragged along the things associated, the things which are linked to it. So therefore, in that sense, we have lost the genetic diversity because of the selection at this locus. Okay, so this is a hard sweep because, again, remember, this green one could have been lost. So as I've been emphasizing, so you might find it weird that why is she saying that, or look at only those stochastic trajectories in which the fixation actually occurs. So this is why I'm doing it, because after the end of the day, after all this has happened, this is the data I will get. I'm not going to look at, I will not get the data in which this came and lost. This is the data I will see, right? Okay, so these are the trajectories which are available to us where fixation actually occurred. Okay, and the time which has taken to go from here to here, it's not 1 generation, of course. This is our friend, the fixation time. Okay, so this is more properly the conditional mean fixation time. Okay. This is what I have been spending some time on stressing. Okay, so that's basically your this very simple limits. This is basically the hard sweep. But of course, you can have mutations. Recombination could, now I will turn it on. Okay, and selection could also be not necessarily be very strong. All these things have been studied a lot with people who are sort of the leaders in the subject, and you can do very simple, not simple, deterministic theory, Maynard, Smith, and Haig, as I said. Let me write it, Maynard, Smith, and Haig, as I said already. So they have done a deterministic theory, but stochastic theory has been also considered by Stefan and others. And simple arguments can also be given if you're very used to thinking about them, by Nick Barton. But there is a lot of literature, and it's, it takes a while to sort of understand how to get these answers quantitatively. So from whatever we have learned using that, I will just tell you, I'll try to answer this question, that suppose suppose we go to something very simple, just 2 loci, because we just learned how to do linkage, how to understand recombination with 2 loci. So again, we go to just 2 loci problem, 2 locus problem. Okay, and I'm just asking that, let's say this is a selected locus. That means it has this B1 and B2. B1 is neutral and B2 is selected. It came a new mutation, remember? And this is a neutral locus. So if B2 comes and it manages to not get lost, and let's say initially P2 came with A1, so you expect that mostly A2 will be lost. So we'll get mostly A1, B2 combinations, right? With some recombination, A2 can also come, but mostly there will be a big change in the frequency of A2. And that's the quantity that one can try to understand what that is. So it's a 2-line argument. Let me give it to you. So I want to know what's the change in frequency of A2 over the sweep period. And just take my word, is proportional to the LD. Why? Because if LD is not there, if they are in equilibrium, as it would happen when this recombination is large, then of course, this one, our mutation did not get much time to drag them, right? Because recombination was large, so therefore there was all these breaks happening here, and it need not remain associated with them, and therefore not affect neutral diversity. On the other hand, if recombination is small, and in fact, we just saw that there was no recombination here, then it will remain associated, and therefore we expect a large change in the frequency. Okay, so this comes again from more detailed calculations, but you can make sense of this, that this change is proportional to the linkage disequilibrium, which we said is e to the power minus r t fix, because that's our sweep period. And this, again, we wrote, I had written this in box. It was 1 by s log of ns. So that's it. That's the formula. So it's saying how much time, so how much is the change in the neutral diversity in this simple, this two locus model is given by this sort of formula. Those who are not happy seeing both the things together, I'll rewrite. It's given by this. Okay, so it looks a little complicated, but it's not so bad. And it's just saying basically the competition between two time scales here. One is the fixation time versus recombination time. These are the two time scales. If the time to recombination is long, that means things remain associated, longer compared to T fix, then there would be a large change in the neutral diversity, otherwise not. So it's basically founded on this argument is founded on the competition between these two time scales. Okay, and if anybody is interested, they can look up more detailed calculations of the diffusion theory with mutations, recurrent mutations, and there's a lot of data, and people have been, of course, looked at it and see and ask, whether these numbers match, and more sophisticated models, not just with two loci. And I just didn't bother to, get all those slides together, but I encourage you to look at, there are several review articles also, which you can look up if this is something you find fascinating. Okay, but coming back to a point, namely, can neutral diversity be reduced? The answer is yes. Okay, and can it be reduced sufficiently? That remains still an open question. Okay, so the last thing I want to say is, let's finish this point. So I just spoke to you about beneficial sweeps. So one more point I want to make. Okay, I just discussed that with you. But if this mutation was not beneficial, suppose it was deleterious, and although we agree that if it is strong, it's unlikely it will fix. Okay, but suppose it's moderately strong or weakly deleterious, there is a chance for it to fix. Similarly, if this one is a neutral one, okay, it's a new mutation and it's neutral, it can also fix. There's a chance for it. Then also there's a reduction in neutral diversity. And however, the neutral sweeps and deleterious sweeps are not that well studied. Okay. So the reason being that, mostly they can also occur, but they're not really that well studied. The reason being that they will not fix, but what one forgets is that deleterious mutations and neutral mutations are much more common than the beneficial ones. Beneficial will come, you remember DFEs, the beneficial part is tiny. Most mutations are deleterious and neutral, and they'll keep raining down and can contribute to the sweeps. Okay, so these can occur, but they are not very well studied. And what is their contribution to the evolution, to the evolutionary process, the diversity that we see? That is not still clear. I mean, it's an ongoing work. Various people are interested in it. So, but it's still in sort of an open question. It's not, we probably expect that maybe it will not make much difference, but it affects some other things. Okay, so which I'm not telling you what they are. But again, if you are interested, let me know. I'll send you the literature. Okay. But let me just say one more last thing here for deleterious sweeps. I said the fixation time is going to enter for the diversity, right? So it was e to the power minus r times t fix. But I also told you that because of the Maruyama Kimura symmetry, this t fix is same for both positively and negatively selected mutation. right? So these two times are same, which means I will see the same footprint, the same reduction, amount of reduction would happen, whether it's a beneficial sweep or deleterious sweep. So they, so they will leave the same footprint. And that's the sort of worry that, we are saying this is beneficial sweep and we are not studying deleterious sweeps very well, right? But how do you know this is due to beneficial sweep and not deleterious one? Because the numbers are same, because there is a symmetry. Okay, so as I said to you, the symmetry plays an important role in the sense that we cannot distinguish between these two types of sweeps. Okay, so I'm really going slow. Okay, so let's now come to the last point, which is about background selection. Let me say it very quickly. So what is background selection? So we spoke about beneficial sweeps, and I said deleterious and neutral also possible, but deleterious mutations are really important because there are many more of them. And what Brian and others discovered is that that can also lead to the loss in genetic diversity. So let me explain that point. So suppose again, we take our 5 individuals. Okay, and we have our 5 sequences. Okay, and as before, we have this various diversity, various types of neutral sites. Okay, and now deleterious mutations appear. And because deleterious mutations are more common, it's not like there's only one will come, several can come, several can be there, right? Let's take, for argument's sake, that assume that these are strongly deleterious mutations. Then you know that they will not fix, of course, but they will get lost, right? So there's no recombination. Then what would happen? This will get lost, right? Because it's strongly deleterious, its fitness is so small, and fitness of this one is so small that it's unlikely that they will form the next generation or succeeding generations. So in the succeeding generations, these sequences will not figure. And then, therefore, as a result of that, you will have sequences like this. So maybe these ones come back. Okay. So I'm drawing the same way, same color. Okay. But sorry, this one is lost. So we are still assuming that we have 5 sequences, but this particular one is lost. The diversity linked to these kind of sequences will be lost. Okay, so this was not correct. Sorry. So we'll have some other ones. So maybe this one gave rise to that, because we are assuming, let's say, the population size remains constant. Okay, so in that sense also, this also reduces the genetic diversity, the neutral genetic diversity. So again, one can calculate heterozygosity, neutral diversity, etc. But remember, because they are deleterious mutations, we cannot do a simple theory. Here we have to do somewhat more complex ones, and this has been done using at least 2 approaches. One is using coalescent, which we did not discuss at all, and so that's Hudson and Kaplan. I don't have it here, perhaps. In 94, and then Charles et al. Yeah. Go ahead, Bhavya.
Yeah. So in your example, the mutation.
Bhavya, your voice is breaking. Can you repeat?
Hello. Can you hear?
Yeah.
Yeah. I read mutant background. So, possible that in one background it stays, in other the background selection happens and it gets sweet.
That these red ones could sweep? Yes, sure, it can happen. But I'm saying that, so that's why the deleterious sweep, we'll call it deleterious sweep. That's one mechanism that will also reduce genetic diversity. Okay, right? Just like we said for beneficial sweep. But I'm saying the fact that there are a lot of deleterious mutations which are strong, and therefore they are more likely to be lost than sweep. The more dominant mechanism is background selection and not deleterious sweep. I'm not sure if you were asking this question because I couldn't hear you very well. I'm just saying that if it was mildly deleterious or moderate, yes, we should talk about deleterious sweeps. But these are the ones which are strongly deleterious and they are more likely to be lost. And therefore, when they lost, that means they're removed from the population. Okay. And that's how they will take out the genetic diversity away. That's how they will sweep it away because they are themselves removed. They cannot form the next generations.
Yeah, my question was whether the background itself in which those deleterious mutations are there would matter.
Yes. This is the background in which there is some linkage, linked loci with a deleterious mutation.
Yeah.
So
I mean, what I was saying is here there are two different backgrounds, right? So
yes.
So
I'm saying that each of them is pretty bad, and I'm putting two of them, unlike the beneficial case, because deleterious mutation, several of them will actually carry this red mutation, because deleterious mutations will reign more often. No, that's why I wanted to emphasize that. And that's why I have put several of them. In fact, afterwards, then when these guys are lost, you will have more. They will keep coming. This is the recurrent mutations. For deleterious one, single mutation will not do much. The key point, which I should have stressed, is that there are recurrent deleterious mutations. Then only they make a difference. They'll keep happening. After these two are lost, new ones will come on, for example, some other background. And this will keep happening. And the sum total of this whole effect at largish times is a reduction in the diversity.
Okay.
Yeah. Okay, so please ask me later. I think I'm running out of time. So let me just finish with the last couple of things. Okay, so again, one can do more, detailed theories, but I will just say that, okay, I'll just give you the answer, I think. So one can find this diversity loss. It turns out to be something like, because you'll see this formula sometimes. So if S is the selective effect of the deleterious mutation, and R is the recombination rate, and mu is the mutation. So mutation did not enter for the beneficial case. I, of course, neglected, but mutation is really important here because it's the recurrent mutation which matter. Okay, so there is some formula like this, which you will get, but it requires a bit of work. Okay, so fine. So this one can do and so on. And one can also think in terms of effective population sizes. But let me not start on that. Okay, so I think I'll just stop here by just giving you an outlook of things that I wanted to, sort of do. So these various, at least three of this mechanism that I have to describe to you, they all help in, reducing our, like, for example, effective population size, and therefore allowing us to make contact with the Kimura's answer. But remember, first of all, any is not always possible to write. Okay, so whenever we can, it's sort of going mostly in the right direction. Okay, so however, even if we take these factors into account, there is still several orders of magnitude discrepancy that remains. So we still don't understand, like, exactly quantitatively, we still don't understand how this genetic variation is, what kind of factors determine it and how much do we need it, and so on, which won't fully understand it. And I think one reason is simply because most of the work that has been done so far assumes that it takes each of these processes separately, because these are complex processes, very difficult to make sense if you put all of them together. So now people are now sort of starting to understand the effect, the joint effect of one or more of these processes, for example, demography and selective sweeps. Okay, so the joint effect, and then this is of course more complex and not so easy, but that's the sort of one work going on, and so we are also interested in this, and I'll probably describe some results next week. And although I said that Lamontin's paradox was the running thread through these 3 lectures, I wanted to make this story and present this to you. Each of these directions, I mean, so like 3 mechanisms that I said, each of them, however, has its own story. So I did not, for example, delve in detail about the sweeps, but itself, it's a, you know, explosively growing field. So even if you don't are so much interested in the paradox of variation, within sweeps itself, there's a lot of work to do. Okay. Similarly, for mutation rates, we don't know how mutation rates evolve. Recombination rates, I did not speak much about, but how do recombination rates evolve? And those insights can feed back into, again, understanding this paradox of variation. So in my group, we are interested in all these problems in the sense that we are interested in mutation rate evolutions. I have worked on that before, but I'm still interested. And next week, I will describe some of our results on diversity, how that is affected if we take changing environment into account. Okay, so I think that's all I have to say. So I'll stop sharing now, unless there's a question. Are there any questions? Let me just stop here. Okay.
Hi, yeah, I actually have a question.
Yeah.
So this was about the Maruyama Kimura symmetry that you spoke about. Yeah, so I thought it was very surprising. And you said there was the conditional mean time for fixation that is symmetric.
Yes.
And so do we know what happens to higher moments?
Good question. Those are hard actually. I don't know. Sachin, do you know?
I mean,
for the variance?
Yeah. So
yeah, I don't.
Yeah, the distribution is in fact.
No, I don't know.
We
now.
haven't calculated yet.
No. Actually, it's a good question. Yeah, I don't know. Yeah, that's a nice question. Yes. Yeah. I mean, at least one should know about the variance. I think we can. No, I think I did check it. In fact, yeah, without dominance, I think it happens for variance as well. I'll check it and let you know. If you can just send me your email address or write to me, I will let you know. But thanks for the question.
Sure. Yeah. Thank you.
Okay. Any other question? Okay. All right. Okay. So thank you very much. And I enjoyed the discussion. And thanks for listening. And I hope to see you guys for the later lectures as well. Okay. So I think we start at 2 o'clock. In fact, you guys will be telling us more about your posters. So see you then.